Imagine this, something incredibly tiny, a microscopic fungus, just living unseen inside the grass, the very grass that, feeds so many animals. And this little hidden helper gives the plant amazing resilience, helps it fight off drought, heat, pests. But here's the twist, the real paradox. That same microscopic helper, it can become a silent killer for the animals that rely on that grass. So today we're going to plunge into this fascinating and frankly pretty dangerous world of endophyte mycotoxicosis. And for this deep dive, we're not just talking about, say, cattle or sheep. We're really zeroing in on two groups that often get overlooked but have really distinct reactions. The Camelidae, the llamas, alpacas, camels, and the Cervidae, that's deer and elk. Our goal today, to kind of cut through the science jargon and give you, whether you're in specialized farming, managing animals in a zoo, or working in wildlife conservation, some really crucial insights into this threat. It's widespread, but maybe underappreciated.
Yeah, and what's really striking is exactly that dual nature you mentioned, that fungus-grass partnership. It's a genuine biological double-edged sword. See, the very compounds, the alkaloids like ergovaline and lolitrim B produced by these fungi, they're what give the grass those great farming traits, drought tolerance, pest resistance, all that good stuff. But those exact same compounds are what cause these really severe, sometimes fatal diseases in the animals eating them. We'll mostly be looking at two main types of toxins today. First, the ergot alkaloids. Ergovaline is the big one in tall fescue. It's a powerful vasoconstrictor, tightens blood vessels. And second, the indole terpenes like lolitrim B. You find that mainly in perennial ryegrass, and it's a potent neurotoxin. Causes nerve issues. But, the symptoms, the clinical picture, it's not straightforward. It really depends heavily on the animal species, its health, its age, the environment. And that variability is exactly why we need to dig into how these different groups react, especially the llamas, alpacas, and deer we're focusing on.
It's just incredible, isn't it? Something so basic for the plant's survival being so harmful really highlights that hidden world right under our feet. So what exactly are these organisms, these endophytes that manage this tricky balancing act?
Well, they're fungi, specifically from the Epicloe genus, and they form this really deep mutualistic relationship with common grasses. Think tall fescue, especially that Kentucky 31 type you hear about, and perennial ryegrass. Basically, the fungus gets a safe place to live and nutrients from the grass. And in return, the grass gets these amazing competitive advantages. It can handle drought, heat, poor soil. It fights off insects, nematodes. That's why there's something like, 35 million acres of this infected, often toxic fescue just in the U.S. It's a very successful survival strategy for the plant.
Wow, 35 million acres. That's a huge area. It really drives home the scale of potential exposure for livestock and wildlife. But you mentioned how it helps the grass compete. Does this fungus spread easily between plants in a field? Or is it more contained? Thinking about how you'd manage it.
That's a really critical point for management, actually. No, it doesn't spread like, say, rust fungus between mature plants in a pasture. It's transmitted vertically. That means it passes directly from the mother plant to its seedling only through infected seed. And this vertical transmission is absolutely key. It means your control point, your main strategy for long-term management has to be the seed source, not trying to stop it spreading plant to plant in an established field.
Okay, that clarifies things a lot. Vertical transmission through seed. Got it. So let's get a bit more specific on the toxins themselves. You mentioned the two main classes, ergot alkaloids and indole terpenes. Can you elaborate a little more on those? And crucially, where in the plant are they most concentrated?
Right. So the first class, ergot alkaloids. In tall fescue, the main one we worry about is ergovaline. Now, it's related to the stuff that causes classic ergotism from Claviceps fungus, but it's distinct. Ergovaline's big effect is potent vasoconstriction. Then you have the indole terpenes. Lolitrim B is the primary one here, found mainly in perennial ryegrass. This one's the neurotoxin. It's what causes that condition called ryegrass staggers.
Look.
Now, thinking practically, where are these toxins highest? They aren't spread evenly. Generally, you find the highest concentrations in the seed heads and down near the base of the plant, in the leaf sheaths, the actual leaf blades, usually lower concentrations. This is super important if you're trying to manage grazing. And other things can boost toxin levels too, like heavy nitrogen fertilizer use or environmental stress drought, high temperatures. That really helps explain why that summer slump you see in animals grazing, toxic fescue gets so much worse in the hot, dry summer months. The plant's stressed, making more toxins.
Okay, that's a very practical link between environment, plant stress, and toxin levels. Really useful. So before we really get into the unique ways camelids and cervids react, which sounds like where things get really interesting, maybe let's quickly set a baseline. How do these endophyte toxins usually affect the more commonly studied animals, like cattle and horses, just so we appreciate how different the others are?
Absolutely good idea. Let's take cattle. The effects there, mostly from ergot alkaloids and fescue, are pretty well known. You get fescue foot, that's awful. It's a gangrene of the feet, tail, ears, usually in cold weather because the blood vessels are so constricted. Then in hot weather, the opposite problem, summer slump. Same vasoconstriction stops them cooling down properly. So, hyperthermia, poor weight gain, rough coat. Less common, but nasty, is bovine fat necrosis, where hard lumps of fat form inside the abdomen, can cause blockages, often linked to heavily fertilized pastures, and yeah, general reproductive problems too. Now, horses, they are exceptionally sensitive, especially pregnant mares. It's really quite bad. Ergot alkaloids can cause just devastating reproductive failure. We're talking prolonged gestation mares carrying foals way too long, agalactia, no milk production at all after foaling, dystocia, which is difficult births, thickened placentas, and tragically higher rates of foal death. If you have breeding horses anywhere near fescue, it's a massive management headache. And then shape. Compared to cattle and horses, they generally handle the ergot alkaloids in fescue a bit better, more tolerant. However, they are highly susceptible to lolitrum B, the neurotoxin in perennial ryegrass. Ryegrass staggers is a really big deal for sheep in places like New Zealand, where ryegrass is common pasture, so you see. Even among the usual domestic animals, there's quite a range of symptoms and sensitivities depending on the toxin and the species involved.
That baseline really sets the stage. Fescue foot, summer slump, reproductive disasters in horses, staggers, and sheep. Okay, now let's pivot to our first unique group, the South American camelids, llamas and alpacas. You mentioned they're pseudo-ruminants, different gut system, and they evolved mostly away from these specific toxic grasses. I guess that lack of evolutionary exposure makes them more vulnerable. What does that vulnerability look like in practice for, say, an alpaca farmer?
You hit it exactly. Their evolutionary history likely plays a huge role. And what's really striking, clinically striking, is their exceptional susceptibility to lolitrum B, the neurotoxin from ryegrass. This leads to a really severe form of ryegrass staggers. Anyone managing llamas or alpacas needs to recognize the signs because they're quite dramatic and worrying. You'll see tremors, especially head and neck tremors first. Then it quickly progresses to ataxia, that serious lack of coordination, a very stiff, stilted way of walking, staggering, falling over, even convulsions in bad cases. And here's a critical thing. These signs get way worse with any kind of excitement or if you try to move them. Handling becomes incredibly stressful and risky for the animal. We also generally see that younger animals, crias, weanlings, they tend to be hit harder, more susceptible, more severe signs. And sadly, if they're exposed for too long, it can cause permanent brain damage. But don't forget the ergot alkaloids either. They are susceptible to ergoline from fescue too. That shows up mainly as really significant heat stress because of the vasoconstriction, which is a big problem in warmer areas. Plus weight loss, poor condition, just general ill thrift. And we have actually seen documented cases of fescue foot-type lesions in camelids to sloughing skin on extremities. Confirms that severe vasoconstriction happens in them as well, maybe even more acutely than in cattle. And yes, reproduction suffers too from ergot alkaloids. Lower fertility, pregnancy losses, poor milk production, agalactia again, which fits perfectly with that prolactin suppression mechanism we talked about.
Wow. So for camelids, it's severe neurotoxicity from ryegrass and significant heat stress and circulatory problems from fescue. That's a double whammy. Okay, that's a really clear picture for camelids. Now let's switch gears to Cervidae deer and elk. You said their story has a particularly startling, often fatal twist. Given how widespread toxic tall fescue is, especially in the U.S. fescue belt, lots of wild and farm deer are definitely exposed. So what makes their reaction so different, so alarming?
Yeah, this is something that I think really needs more attention, especially in wildlife management and potentially even zoo settings. The most severe and critically often fatal issue we see documented in cervids is this unique condition called abdominal lipomatosis. Basically, it's severe fat necrosis, but in a very specific and deadly pattern. We have case studies, particularly from farmed Eld's deer, describing these massive, incredibly hard, almost rock-like fat deposits forming inside the abdomen. And these fat masses literally grow around and constrict vital organs. Specifically, they often block the ureters, those tubes that carry urine from the kidneys to the bladder. So urine backs up, causing severe kidney damage that's called hydronephrosis, and it eventually leads to terminal kidney failure. And the evidence strongly links this directly to chronic exposure to ergovoline from toxic tall fescue. It seems to be a very specific metabolic response in deer. It's completely different from the fescue foot or summer slump you see in cattle. So for anyone managing deer, wild or farm, this is a huge challenge. An animal might just look like it's losing condition or acting off, but the underlying cause could be this silent, progressive, fatal organ compression. You might be looking for the wrong thing entirely. And think about the bigger picture for wild deer populations. The implications are profound, but honestly largely unstudied. We're talking potentially reduced body condition, maybe impaired reproduction, definitely increased vulnerability to heat or cold stress because of underlying metabolic issues, possibly even changes in where they forage to avoid toxic areas. It's a major, major knowledge gap in wildlife toxicology right now.
That abdominal fat necrosis, it's truly shocking. Just constricting the organs from the inside out. It makes you wonder how. How do these microscopic fungal compounds, designed to help a plant, end up causing such specific and devastating damage in different animals? What's happening at the molecular level? Let's get under the hood.
Okay, yeah, the mechanisms are pretty fascinating and quite different for the two main toxin types. Let's start with alkaloids, like ergoline. The core issue is that sustained vasoconstriction, the tightening of blood vessels. How does it happen? Well, ergoline is a clever mimic. Its molecular shape is remarkably similar to some of our own key neurotransmitters, serotonin, dopamine, norepinephrine. These guys regulate all sorts of things, including blood vessel tone. So ergoline basically hijacks the system. It binds to the receptors meant for those neurotransmitters, specifically alpha-adrenergic, dopaminergic, and serotonergic receptors on the smooth muscle cells in blood vessel walls and also in the brain. Depending on the receptor, it can either stimulate it or block it, but the overall effect is persistent vasoconstriction. Blood vessels stay narrowed, blood flow is reduced, and the consequences flow from that reduced blood flow. Impaired thermoregulation is a big one. Can't cool down in summer slumps. Can't keep extremities warm in winter, fescue foot, frostbite. Reduced blood flow over time can also lead to tissue death, necrosis, gangrene. But there's another key mechanism, especially for reproduction. Ergot alkaloids are strong agonist stimulators of D2 dopamine receptors in the pituitary gland. This strongly suppresses the release of a hormone called prolactin. Prolactin is vital for milk production and plays roles in regulating pregnancy cycles. So suppressing prolactin, that's why we see the agalactia, no milk, the prolonged gestation, especially in mares, and even things like retaining the heavy winter coat into summer, which makes heat stress even worse. Okay, now switch gears to Lulitrim B, the neurotoxin from ryegrass, totally different target. Its main target is a specific type of ion channel found in neurons called large conductance calcium-activated potassium channels. You might see them called BK channels or MaxiK channels. Think of these channels like crucial off switches or reset buttons for nerve cells after they fire an impulse. They help the neuron get back to its resting state quickly so it can fire again properly. Lulitrim B basically jams these channels. It inhibits them, so the neuron can't reset properly. It stays overly excitable, fires erratically. This leads to hyperexcitability throughout the central nervous system. And that's what causes the clinical signs, the fine muscle tremors, the severe incoordination ataxia, the staggering walk, the inability to sense limb position properly. And remember how we said signs worsen with excitement? That makes perfect sense now, right? If the neurons are already on edge, any extra stimulation just pushes them over the limit.
Wow. Molecular mimicry for vasoconstriction and jamming the nerve's reset button for the tremors. That's a brilliant explanation of how they wreak havoc. But okay, given these varied signs, heat stress, lameness, neurosigns, or even just subtle weight loss hiding that fat issue in deer, how do you actually diagnose this? How do you figure out if endophyte toxicosis is the culprit? What's the practical challenge?
It's definitely a challenge sometimes. It really requires a combination approach, almost like detective work. The absolute gold standard for confirming exposure is testing the forage or feed itself. We use a lab technique called high-performance liquid chromatography, HPLC, usually with fluorescence detection because it's very sensitive. It involves carefully collecting representative samples of the pasture, hay, or feed the animals are eating. Then it's dried, ground up, the alkaloids are extracted and purified, and then the machine quantifies the exact amount of ergovaline, Lulitrim B, or other relevant toxins, usually down to parts per billion, PPD levels. That tells you definitively if the toxins are present and, crucially, at what concentration. Then, on the animal side, you combine that forage data with clinical diagnosis. This means looking at the whole picture, getting a good history. Were they grazing fescue or ryegrass pastures known or suspected to be toxic? Observing the clinical signs. Are they showing those characteristic neurological signs like tremors and ataxia we see in camelids? Or maybe the nonspecific signs like poor growth, weight loss, maybe signs of kidney issues in deer? So again, diagnosing that fat necrosis in deer before death is really, really tough. And it's absolutely critical to exclude other diseases. Lots of things can cause tremors or weight loss. You have to rule out parasites, other neurological conditions, nutritional deficiencies, etc. Sometimes a necropsy, an animal autopsy, is needed for a definitive diagnosis. That's where you'd clearly see the fat masses in a deer or maybe characteristic brain changes in a camelid with chronic staggers. And this whole diagnostic puzzle leads us to a really critical area: toxicity thresholds. We need to know how much toxin is too much. But this is where we often lack solid data, especially for camelids and cervids. For cattle, yeah, we have general guidelines. Maybe 100, 500 ppd or govaline starts causing problems. For lowly Trem B, maybe 800, 2000 ppb triggers staggers in sheep or cattle. But this is vital. These thresholds are highly species specific. You cannot just apply cattle numbers to alpacas or deer. Remember that camel study I mentioned? It suggested a safety threshold of maybe 500 ppb, low term B for camels, and needing to dilute feed if it hit 1,000 ppb. That's way lower than the threshold for cattle or sheep. It just hammers home the urgent need for species specific research. We're likely underestimating the risk for camelids and cervids by using thresholds derived from traditional livestock.
That lack of specific thresholds is a really big gap, isn't it? Makes managing risk much harder if you don't know the exact danger level for your animals. So, okay, bringing it all together, we understand the toxins, the mechanisms, the varied symptoms, the diagnostic challenges. What does this all mean for you, the listener, if you're managing these animals? What are the practical steps? What can actually be done to protect llamas, alpacas, deer, elk from this hidden threat?
Right, it really comes down to an integrated management strategy. There's no single magic bullet. The most definitive long-term solutions usually involve agronomic and pasture management. This is tackling the source. Option one is eradication and renovation. That means killing the existing toxic pasture and replanting with non-toxic options. There are novel endophyte fescue varieties now that give you the good agronomic traits, drought tolerance, etc., but without the harmful toxins. That's the ideal, but it's expensive and labor intensive. Another approach is dilution. If you can't renovate, you can overseed the toxic pasture with non-toxic plants, especially legumes like clover or alfalfa. This reduces the percentage of toxic grass the animals consume in each bite. And then there's smart grazing management. Remember how toxins are highest in seed heads and the base? You can manage grazing to avoid those parts. Mowing pastures before they seed or using rotational grazing to keep the grass taller, say 3-4 inches minimum, forces animals to eat the less toxic leaf blades. Then you have strategies focused on the animal and herd management side. Strategic pasture use is key. If you have safer, tested pastures, move your most susceptible animals there. That means late gestation females, young growing animals. Keep them off the high-risk stuff. Supplementation or diet dilution also works. Providing other feeds good quality hay that's tested clear, or grain supplements physically dilutes the amount of toxin they ingest relative to their total diet. People also try toxin binders. These are feed additives, often clay-based products or yeast cell wall extracts. The idea is they bind the alkaloids in the animal's gut, preventing absorption into the bloodstream. The scientific evidence on their effectiveness can be a bit variable depending on the product and situation, but some people find them helpful. And supporting the animal's overall health with antioxidants like vitamin E and essential minerals like selenium, zinc, copper can help them cope with the oxidative stress the toxins cause. And finally, there are veterinary interventions for animals already showing signs. The absolute first, most critical step is removal from the source. Get them off the toxic pasture or feed immediately. Many early signs, especially the neurological ones from lowly Trem B, can be reversible if exposure stops quickly. Then it's supportive care, providing shade and plenty of cool water for heat-stressed animals, maybe using supportive slings for animals down with severe staggers to prevent injury, just helping them through the crisis. As for specific drugs, there isn't really an antidote that neutralizes the toxin itself. For the reproductive problems in mares caused by fescue, veterinarians sometimes use dopamine antagonists like domperidone to try and counteract the prolactin suppression. Whether that works well in camelids is less clear, needs more study. So mostly it's about stopping exposure and supporting the animal until the toxins clear their system. So I think what we've really highlighted today is just how different endophyte toxicosis looks in these specific groups. Camelidae, you've got that extreme sensitivity to neurotoxins causing severe ryegrass staggers. Cervidae, you have this unique, often fatal abdominal fat necrosis from fescue. And this raises, I think, a really important point. You simply cannot assume that what happens in cattle or sheep will happen in llamas or deer. You can't use the same diagnostic clues or expect the same outcomes. Ignoring these species-specific differences, it leads to real economic losses, sure, but more importantly, it creates significant and often completely overlooked animal welfare problems.
That's a really crucial point to end on, the danger of extrapolation. So what does this all mean for you listening right now? Whether you're running an alpaca farm, managing deer at a park, working in conservation, it really underscores, I think, an urgent need for more research. We desperately need solid, species-specific toxicity thresholds for camelids and cervids. We just don't have them. We need proper studies looking at the impact on wild deer and elk populations. Is this toxicosis a hidden factor driving population health or dynamics? We barely know. And we need to understand the why. Why the unique pathophysiology? Why do camelids get the staggers so badly? Why do deer develop this fatal fat condition? Understanding the mechanism is key to better diagnostics and maybe treatments. And of course, validating treatments that actually work for these specific species. It's amazing, really. This tiny fungus, hidden away inside a blade of grass, forces us to constantly rethink how we understand animal health, right across the board. The deeper we dive into these relationships, the more we uncover these unseen forces that are shaping the lives and welfare of animals, both on farms and out in the wild. It definitely leaves you wondering, doesn't it? What other hidden symbiotic relationships, maybe even right around us, are having profound impacts that we just haven't recognized yet?